cellular fluid, ito yung area within the cell o ito yung cytoplasm. So, the membrane will serve as the barrier. So, not all, mostly lang, selective na molecules can enter the cell. But based on the entry of cell in and out of the cell, there are several membrane transport activity. So, cellular membranes are fluid mosaic lipids and proteins as described by most scientists. So, phospholipids are most abundant in lipid in the plasma membrane. And phospholipids in the most plasma membrane are bilipid in layer. So, they are amphipathic molecules containing hydrophobic and hydrophilic regions. Okay, cellular membranes are fluid mosaic of lipids and proteins. So, majority the plasma membrane is composed of phospholipids, a certain type of lipid. And it consists of they are amphipathic molecules containing two ends, the hydrophobic and the hydrophilic regions. When we say hydrophobic, this repel water, it is not soluble to water, and hydrophilic, it attracts water. So that those are the regions of the phospholipids. And why is it called fluid? Fluid mosaic model states that a membrane is a fluid structure with mosaic of various proteins embedded in it. If we are going to see the structure of the membrane, have the portion of the membrane. So this is a phospholipid bilayer. So when you say bilayer, there are two layers of phospholipids aligned of so tawag niya sa arrangement arrange tail to tail. So the hydrophilic head, so this is the hydrophilic head which attracts water, and the hydrophobic tail doesn't attract water. So kaya not all molecules can enter because of the polarity also of the membrane. And who invented the theory of mosaic model of the plasma membrane? In 1935, Hugh Davison and James Danieli proposed a sandwich model in which the phospholipid bilayer lies between two layers of globular proteins. And later, based on the study, my problem with their model. So, Singer and Nicholson proposed that the membrane is a mosaic of proteins dispersed with the bilayer. So, when we say mosaic, they are not uniformly distributed. Para silang lumulutang sa sea of phospholipids. So, muna ang imagination nila Singer and Nicholson in 1972. So, this is how Nicholson and Singer presented what is a plasma membrane. So, we have here the phosphobilipid layer, and the proteins these are globular proteins that are embedded in the sea of phospholipids. Some proteins are only embedded superficially, some are in between the bilayer or transecting with the bilayer. So, para siyang sandwich. Okay, hydrophobic region of the protein and the hydrophilic region of the protein. And these proteins are helpful in transporting molecules in and out of the cell. Freeze fracture studies in a plasma membrane support the fluid mosaic model. So between this, the proposal ni Singer and from the two scientists, through this method, it proves that the proposal or the model of Singer and Nicholson. So freeze fracture. So this is the technique that splits a membrane along the middle of phospholipid bilayer. So this is the technique in which they have to parang i-freeze nila yung cell. So they have also to use electron microscope to view the structure of the membrane. So it is not an ordinary compound microscope. It is a high graded, 'di ba sabi niya SEM yung uses electromagnetic kuan na in aside sa lenses, it already use electromagnets. So this is the structure of the membrane. So they slice the plasma membrane in two layers. So na-divide nila in two layers. And they saw yung mga proteins embedded in between the phospholipid layer. So the result muna inside of extracellular layer, and this is inside of the cytoplasmic layer. So inside of extracellular and yung intracellular. So the fluidity of membranes. Phospholipids in the plasma membrane can move within the bilayer. Actually, they are kung ano sila, kung made up of organic compounds, para lang nagkatabi lang ito. Hindi talaga totally parang sa mga covalent bonds and ionic bonds, nagkatabi lang ang mga molecules nito. Most of the lipids and some proteins drift laterally. Rarely does the molecule flip-flop transversely across the membrane. So, para lang sila nagkatabi. So, we have another compound that attaches to the membrane. We have the glycolipid. Mura siya carbohydrate at the same time lipid. And we have this, the protein. And not all the time the proteins are transecting the bilayer. Some are only superficial. So the proteins that are passing through the two layers are called integral protein. And the proteins that attach only to the surface is peripheral proteins. And the proteins that contains glucose or a carbohydrate, we have the glycoprotein, and a compound that attaches a carbohydrate attaches to the phospholipid is called glycolipid. Okay, yun yung mga terms. So these are the lateral movement occurs 10 nanotimes per second. So this is the flip across the membrane. So sa cell biology na ninyo, tunan. Okay. So as the temperature cools, the membrane switch from fluid state to a solid state. So since naa siya semi-fluid siya, so when the temperature drops, it goes to solidification. So mura pud kuan water, pero depende lang sa characteristics. So ang nagre-react tayo ang ating cells to a certain temperature. So the temperature at which the membrane solidifies depends on the type of lipids. So that is the reason na these lipids keep our cell or avoids our cell to solidify in a certain level of lower, or when the temperature drops to a certain level. Pero may limitation. Kung sobra naman talaga ka negative ang temperature, then that's the time to na magahi na ma-frozen. But like 16, negative 16 or negative or negative 16 frozen na tana. Mga 0 or 0, 1, 2, 3 di pa kaayo, napay certain. Kung mag 12, dili pa ta ma-frozen ana. We always suffer chills. But so that's the reason sa mga lipids, it prevents us to solidify immediately. So membranes in rich unsaturated fatty acids are more fluid than those rich in unsaturated than to the saturated fatty acids. So kung ang membrane daw is rich in unsaturated are more fluid than those in rich of saturated fatty acids. So if the membrane contains unsaturated fatty acids, the characteristics of the cell becomes more fluid. Then membranes must be fluid to work properly. Why? Because this also affects the movement of the molecules, the movement of the vesicles. So, kung solid, the vesicles cannot move properly. Cannot move. Hindi siya makapass through. Kung may fluidity ang dali lang siya makapass through. The steroid cholesterol has different effects on the membrane fluidity at different temperatures, and warm temperatures such as 37 degrees Celsius, cholesterol restrains movement of phospholipids. So, hindi din tayo agad-agad mag-evaporate. So, like, if diba ang law sa liquid, higher temperature, uh, the liquid changes to gas. So, because of these compounds, hindi tayo agad-agad matatransform into, hindi tayo magda-dry up easily. So, the cell will not, this cholesterol will help the cell intact even though the temperature is 37. So, at cool temperatures, it makes fluidity by preventing tight packing. So, Moni and diagram having unsaturated hydrocarbon tails, saturated carbon tails. So, more fluid. Moni ang viscous. Murag, kung viscous pa, murag malapot. Semi-fluid. Semi-fluid. Unsaturated versus saturated. So, muna ang itsura kung saturated hydrocarbon. It becomes more fluid. Magkahiwala-hiwalay ang phospholipids. Pero kung viscous, medyo malapot-lapot, magkalapit ang mga phospholipids. How about kung merong cholesterol with animal cell membrane? So, ito yung itsura kung merong embedded cholesterol. Ang cholesterol kasi magsa-stabilized para in terms of higher temperature, hindi magkahiwa-hiwalay. Kung in terms sa lower temperature, hindi mag-solidify. Okay. Evolution of differences in membrane lipid composition. Variation in lipid composition of cell membrane of many species appear to be adaptation to specific environmental condition. So, nag-a-adjust. Ability to change the lipid compositions in response to temperature changes has evolved in organisms that live where the temperatures vary. So, with the help of these compounds within the membrane, it will help us to adapt. Nag-a-adjust ang cell. So, yun ang mga Membrane proteins and their function. Now, what is the primary role of proteins in the membrane? So, the membrane is a collage of different proteins. Pwede silang magiging site for channel in which they will serve as the channel for the molecules to get in and out of the cell. One example, natay sodium-potassium pump. So, it is made up of protein. Okay, peripheral proteins are bound to the surface of the membrane. Integral, by the name itself, integral, it was embedded between the two layers of the phospholipid. And they are also known as the transmembrane proteins. So the hydrophobic region of the integral protein consists of one or more stretches of nonpolar amino acids, often coiled in alpha helices. So hindi din selective ang channel sa mga integral proteins. It only allows certain specific molecules to enter in that protein. So, muni ang itsura, the schematic diagram nila. Actually, gikan siguro ni sa SEM, tapos they drew it, and ito yung base sa ilang na-observe using the electron microscope kung what is the appearance of the integral protein. So, 6 major functions of the membrane proteins. Number 1, transport, enzymatic activity, signal transduction, cell-to-cell recognition, intercellular joining, and attachment to cytoskeleton and extracellular matrix. So remember those functions. So, muna ang transport. Ito na ay enzymatic reaction. So, kailangan ng enzyme para ma-recognize niya. Then, these molecules will have a reaction. Or, signal transduction, kailangan may receptor, but the substrate must be fitted to the receptor. So, kung ganun, maka-enter a molecule, pero kung hindi siya fitted, it doesn't match its affinity or shape, bakaya, then it cannot go in sa intracellular fluid. Ang iba naman is cell-to-cell recognition. Ang glycoprotein, for example, a protein with a glucose, or we call it a glycoprotein, will communicate with another cell. Situate na yung isang cell may receptor site which fits the molecular structure or molecular affinity ng isang membrane with this glycoprotein. Intercellular joining. So this is an example of intercellular joining. One good example, mga desmosomes. And attachment to the cytoskeleton and extracellular matrix. So those are the 6 functions of protein. Okay, transport, enzymatic activity, signal transduction, cell-to-cell recognition, intercellular joining, and attachment to extracellular matrix. Now, the role of the membrane carbohydrates in cell-cell recognition. Okay, cell recognizes each other by binding to surface molecules. So, like katong from the previous diagram. Some proteins may contain carbohydrates and the extracellular surface of the plasma membrane. So, usually, kaning glycoproteins, ang apex nila is exposed. Yung carbohydrate portion mas exposed sa labas ng cell. So, the membrane carbohydrates may be covalently bonded to lipids, forming glycolipids, since dapat kumbaga naa siya binding site to another protein of another cell or para magka-form ng glycoprotein. So commonly to proteins forming glycoproteins. So mura siya kung may imagine nato ni mura lang gyapon pareha sa enzyme na lock and key. So, katong glycoproteins, katong cell na naay protein na attached to a carbohydrate, makakonect siya sa another cell that has a protein na wala pa'y glycoprotein. So, katong tail, naka-expose na glycoprotein, mo to ang mo-attach sa another protein sa kabilang cell. Pero dapat, ang katong site receptor site ng protein fitted doon sa glycoprotein. Kay kung dili siya fitted, like dili pareho og molecular shape, dili siya pareho og affinity, dili sila magkadikit. So naghapon sa type of bond. So carbohydrates on the external side of the plasma membrane may vary among species, individuals, and even cell types in an individual. So, mo ni nganong daghan ma-infect sa HIV. So this is one example. HIV can infect a cell that has CCR5 on its surface as it most people. Okay. Sa membrane ng maraming tao, we have this type of receptor. Kumbaga, one example siya ng type of glycoprotein sa atin. And unfortunately, si HIV, sa iyang membrane, sa iyang capsule, di siya membrane, kay dili man ni siya living cell, capsulated genetic material lang na siya. Katong capsule niya na nakabalot sa iyahan ay affinity sa glycoprotein na nasa ating membrane. That's why nakakonect siya sa atin. Kasi ang receptor ng CCR5 and receptor CD4 nagkaroon ng affinity kay parang pwede ma-attract, nakadikit, so na attraction, so na affinity, acceptable. Kumbaga sa battery pa, positive o si negative magkuan, di ba? Mag-attract. Kung the same ang polarity, mag-repel. So since ang affinity nila compatible, nakadikit si HIV virus. Ayun. Naka-enter siya because of receptor CD4. Pero kung wala si co-receptor CCR5 sa isang membrane, like for example here, kung wala daw yung isang receptor, ito yung sa left side, ito yung may affinity receptor, ito kung absence ng CCR5, then this virus cannot enter sa system. So, ang HIV virus cannot enter the system. Murag kanong panahon na katong COVID, makita nato na naay mga tao na asymptomatic. Meaning, wala sila nag-exhibit og symptoms but they carry the virus. Murag ngani ang nahitabo kanina sa right diagram. And katong tao na nagkasakit sa COVID-19, ang ilahang mga cells na apektuhan. So, naa sila'y affinity sa virus. So, ang virus nakasulod sa cell. So, mo ni nahitabo sa mga taong nagkasakit sa COVID-19. Synthesis and sidedness of membranes. So, membranes have distinct inside and outside faces. And napos si kanya-kanya mga polarities and affinities. So, the asymmetrical distribution of proteins, lipids, and associated carbohydrates in the plasma membrane is determined when the membrane is built by ER and Golgi apparatus. Kaya asa din nagikan si protein. Ang protein na naa sa cell, giproduce na ni nucleus. Based sa review nato kanina kay Buzman na podcast, 'di ba? Hindi lang nag-contain si nucleus ng genetic material but also a factory ng protein. And iyan ang ipasa kay rough endoplasmic reticulum, then iparepack niya kay para kaning nuclear proteins magamit and mag-transform to another component sa membrane or mahimong another organelle kung naamay na any other use sa protein. So, protein synthesis. So, we have here, mga transmembrane lipids. So, the ER lumen. So, these proteins, kaning small molecules of proteins, actually, they cannot go out directly as a protein. They had to be enclosed by another membrane from the ER. So, kaning muning gitawag na vesicular proteins. So, they have to one direction lang, asa ilang directed direction, they will go to Golgi apparatus. Okay. Once they attach to the Golgi apparatus sa basal part, non-specific part sa Golgi apparatus, they will be again repack. I-repack ulit sila ni Golgi apparatus. So, you have here the vesicle. Pumasok kay the glycolipid. I-ano niya yung protein. Mag-attach siya doon kay protein. Then, pupunta siya. I-repack siya uli. So, si Golgi apparatus may idadagdag. Then, mahimo na silang vesicle. So, this vesicle, pwedeng dito lang gagamitin itong, okay, asa na itong arrow. So, this vesicle, pwede pupunta sa other organelles or they will be released as secreted proteins. So, muna siya. And some will be part sa membrane. So, kaning Y, muna itong glycolipid coming from Golgi apparatus, nagipasakay niya kay protein. So, kaning glycolipid, ipasakay ni Golgi apparatus, katong protein, naghihimo ni nucleus, then, ihanap pong i-repack. Okay, so nahimo na siyang secretory proteins. Okay, mahimo na siyang vesicle, ma-repack na siya. So, kaning Y, represented by glycolipid, kaning violet na lingin, protein, they will go to in the process of exocytosis. So, hinay-hinay ang membrane niya mo attach dito sa plasma membrane. Mo add on siya, but the glycolipid will stay, but the proteins will be released sa extracellular fluid. So, manay nahitabo. So, membrane structure results in selective permeability. So, a cell must exchange materials with surroundings. So, the cell itself needs some nourishment coming from our food that we eat. So, kailangan ang plasma membrane are selective. Hindi din basta-basta because this is also to protect the cell na hindi agad mapuntahan ng mga foreign material or harmful chemicals. Sometimes we take harmful chemicals. We got it from the food, the medicine. Muna ang mga ways na makakuha tag mga harmful substances. And the role also of the protein is hindi agad-agad niya i-accept. Depende kung unsa ang affinity, structure, and its size. So, hydrophobic nonpolar molecules such as hydrocarbons can dissolve the lipid bilayer and pass through the membrane rapidly. Polar molecules such as sugars do not cross the membrane easily. Sugars have transport proteins. Okay, muna itong isa sa mga integral na other type of proteins. So transport proteins allow hydrophobic substances have hydrophilic channel molecules or ions can use as a tunnel or passageway. So channel proteins are also called aquaporins. Facilitate the passage. So this is another type of protein. Now we have another type, the passive transport. So this is the diffusion of substances across the membrane with no energy investment. Pwede lang makasulod, walang hassle. So depende sa concentration gradient. So diffusion is tendency of molecules to spread out evenly into the available space. Pero kung hindi siya mag-fit doon sa space, hindi din siya maka-enter. It also depends on the concentration. It depends on the size of the molecule. Pero kung equal na ang concentration, both internal and external, walang movement sa ganong type na molecule. So let's have an action of a certain membrane for being selective.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions such as sodium ions and hydrogen ions cannot pass through the membrane unaided.
So diffusion is another membrane membrane activity in which the molecules can pass through without hassle. But it depends to two types. We have the passive and the active. Kung passive, simply in and out lang, depending on the concentration gradient. Like for example, kung highly concentrated sodium sa loob, and this will attract water. So the water will move in greater volume compared sa nag-exit na water. So, tendency, maglaki ang cell. Or, kung ang concentration gradient ng solute ay marami sa labas, then the water will be attracted sa solute, since mauna ang nature sa water, then maraming greater volume of water will come out from the cell than the volume of coming in. So tendency, the cell will shrink. Now we have here another video para ma-imagine ninyo what happened to a cell that was exposed onto different types of solution depending on in relation to the concentration of gradient of the cell. We have here hypertonic, in which the cell was exposed to cellular fluid. Yung extracellular fluid ay maraming solute. Ito yung nakapa-attract sa water because water is a greatest so pupunta dahil maraming solute ng cell compared sa labas. So, basically, solute and solvent, maa-attract si water solute nandun sa loob. So, mas maraming tubig ang magpasok. So, tendency, nakita diagram, tumaba si cell. And kung equal naman ang amount of solute sa loob of the para makuan, i-play na natin ang video.
Imagine that you're stranded on a raft in the middle of the ocean. You don't have any emergency food or water, but you also know that humans can only survive about 3 days without fluids. Should you drink seawater or not? Stick with us. We're going to find out and also review hypertonic, hypotonic, isotonic solutions along the way. Cells have a very finite range of conditions in which they can survive. If it's too hot, too cold, too watery, too salty, too acidic, or too basic, they can't function properly. We're going to explore what happens when you place a cell in a solution that is much more salty, hypertonic, much more watery, hypotonic, they allow certain particles to pass through, but not others. Larger molecules can't fit through the membrane without special channels. The cell membrane is fitted with special protein channels called aquaporins, which allow water molecules to cross. It's going to depend on something called the concentration. Concentration is a measure per volume of solvent. In other words, whether the
Pa rin, magkaroon pa rin ng like to reach into equilibrium. Pero iba naman yung sinabi nung host na substance, it depends on the situation. Now, substances diffuses down to their concentration gradient. So, the region along which the density of chemical substance increases or decreases. Transport in which the pass through with so example yung oxygen and water comes through that easily. Transport is osmosis. Osmosis is a diffusion of water across a selectively membrane, just across the membrane from the region. So, dito, set up sa diffusion na simple diffusion, tiningnan yung activity ng how the solute moves. Ngayon, sa osmosis, you going to observe also how the water moves depending on the concentration gradient of the solute. So, water diffuses across the membrane from the region of lower solute concentration to the region of higher solute concentration until the solute concentration is equal on both sides, or we call it the isotonic state. So let's have an experiment. Okay. So let's have this experiment on sugar and water. So the tube containing water and sugar but of different amount of sugar. Sugar will serve whilst water will serve as a solvent since water so mas maganda representation ang water.
Ma'am, nawala na pud ka, ma'am.
So what will happen in a few minutes? Okay, first set up the other side, maraming molecules of na side. Pero dito na side, likely hindi matamis pero may molecules of sugar. If will happen. So, mangyari nito, she will go to the side in which may maraming concentration ng solvent, pero lower ang amount of solute. Seawater pupunta doon sa area na higher concentration of solute. So, if we go back to the definition, from the region of lower solute concentration to the region of the higher solute concentration until concentration is equal on both sides. So, ito yung nangyayari. The water na to work to a region of higher concentration in equilibrium on both sides. So, kanilang diffusion sa water. So, yun yung osmosis. Okay. Not a cell was exposed to different solution. Okay. Tonicity is the ability of the surrounding solution to cause the cell to gain or lose water. So we have three types: isotonic, hypertonic, and hypotonic. So these are the three types of tonicity that's surrounding our cell. Okay, what is isotonic solution? Solute concentration is the same as inside of the cell. So the amount of solute sa loob is equal sa number of solute sa labas. So, what will be the reaction of water? Ano ang behavior ng water sa loob at sa labas ng cell? The water will simply have an equal entry volume of water. At the same time, ang equal din ang volume ng water ang lalabas. Like for example, kunwari, naatay na-enlarge na to ang cell. Tapos, ang isa ka cell, nag-contain siya 50 ml na tubig. Sa labas, na-immerse siya sa 100 ml na tubig. Since equal ang movement equal ang amount of solute sa loob and labas ng cell, meron man din, semi-permeable man siya. So, may lalabas na from the 50 ml, may lalabas na 5 ml na tubig going out the cell. But at the same time, may papasok na 5 ml na tubig sa loob ng cell. So, what will happen sa cell shape? Now, remain lang. Walang nangyari. Kasi the same volume of water ang lalabas, the same volume of water ang papasok sa loob. Yan yung mangyari sa isotonic, sa cell immersed in a isotonic solution. Okay, what will happen to the cell if the environment of the cell is exposed to a hypertonic solution? Like for example, naligo ka sa dagat. Dagat is an example of hypertonic solution. Okay. In a long run, since ang external environment ng cell has a greater amount of solute compared sa loob ng cell, what will be the behavior of the water? Kung kuan pa, si water, large volume of fluid will go out sa cell. So, mas malaki ang lalabas na tubig from the cell going out kaysa papasok sa cell. Okay, for example, let's go back. 50 ml ang content sa cell na tubig. Tapos, immersed also to the same 100 ml na water. Marami man ang solute sa labas. maraming sugar compared sa loob. So si water malalabas sa cell, na-attract kasi siya sa solute. So let's say nawad-an si cell og from 50 ni gawas si ang 30 ml na tubig sa from the cell going out. Pero ang entry ng water going inside the cell is only 5 ml. So, dili equal ang in and out of water. Mas maraming lumabas na tubig galing kay cell compared sa nisulod na fluid going to the cell. So, what will happen to the cell? Magsishrink siya. Kasi wala naman siya, dili man equal ang paggawas sa tubig compared sa pagsulod. So, magsishrink ang iyang size. So, mali siya may tabo. Unsay tawag sa shrinking of the cell? Nato sa first diagram. Plasmolysis. Plasmolysis. Okay, that is okay, muna siya ang reaction sa cell. Now, kung ang cell naman in a hypotonic muna example freshwater. Okay, mga freshwater environment. Munang usahay makita nato kanang mga nalunod, butod. Butod ang lawas. Aside sa nasudlan. Murag ang mga kanang swollen ang mga parts. Okay. This would happen na ang environment outside the cell mas konti yung number of solute. So makita natin dito NaCl, let's have NaCl as the solute. And blue one is ito yung mga tubig molecule, water molecules. What will happen? Anong behavior ni water? Now, ang mangyari, maraming greater amount of volume of water will go in the cell. Opposite sa nangyari dito. Like for example, kung 50 ml ang ang maximum amount na ma-hold sa cell na tubig, instead na ang ilabas niya, maglabas siya ng 30 ml. Dito, magpasok ang tubig 50 ml. Now, what will happen? Cytolysis o cytolysis ang mangyari, magsuswell. But what if kumugo beyond? Muna to ang bursting of the cell or lysis. Naaga po lysis. Bursting of cell. Na tamang term anang bursting of cell. Plasmolysis ang shrinking. Na tamang term. Nakalimot lang yun ko. So, mubuto ang cell. So, mo na opposite reaction sa hypertonic solution. Okay, next. What if kung equal? So, mo na akong ginaingon na equal ang mugawas na tubig, volume sa tubig, like 5 ml ang ligawas, 5 ml lang po ang musulod. So, ang shape ng cell, retain. Walang mangyari. So, ito yung dapat na balance, homeostatic state ng cell within a certain liquid environment. So, muna siya. Muna sa dextrose, dapat unsa ang concentration ng sugar sa dextrose, dapat mo equal siya sa plasma ng nasa ating blood vessel. Kay para ang mga dili maapektuhan ang mga blood cells sa ato ang circulation. Kasi kung sobra pud katamis ang dextrose, mag-create another characteristic sa shrinking is mag-shrink ang mga cells. Pero kung dili mo equal ang solute nag-insert sa bloodstream nato, mulingin ang ato ang blood cell and tendency pwede pa mubuto. So patay ang pasyente ana. Okay, may lang dili mo mag-nurse pero kamo maghihap kung kamo na amay mong doktor, mo direct lang maghihapon mo pila ang concentration ani, i-compute na ninyo. Kaya kung mamali mo compute, mali ang concentration, pag sulod sa pasyente, pag inject sa nurse, mingon na lang ang nurse, dok, patay na ang imong pasyente. So, muna ang dapat ma-familiar, ma-master ninyo ni na action sa cell. Okay. Hope na kanina aspeto you have already understood. So, this with another diagram. Okay. Pero, lahi ang reaction sa plant cell. Plant cell, the cells were not lysed or hindi mag-shrivel because they have this cell wall. So, meron silang cell wall. So, hindi basta-basta puputok. Tapos, hindi din masyado magkaroon ng changes sa shape because of the cell wall. Because the cell wall have fixed shape, will give a fixed shape to the cell. Pero ang plasma membrane, nag-shrink na. Like this one, oh. Ito yung plasma membrane. Nag-shrink na. Okay. So, hypertonic or hypotonic environments create osmotic problems for the organism. Pero kung tanongin ninyo, ma'am, paano nag-survive ang mga isda na nasa dagat? Hypertonic man ang ilahang environment. Naa sila mechanism sa ilang gills na iko-control ang ions, ang solutes na mosulod sa ilang lawas. At the same time, ang ilahang integument mag-secrete og mucus, ang ilang mga scales to protect also from those harsh environment. So, mo po na nakatabang sa mga isda, kana mo nang mo gunit mo sa. So, ang mucus that helps them to keep them from imbalances sa homeostatic regulation. So, that maintains their homeostasis within the body. How about itong mga paramecium, katong mga unicellular organisms na naa sa fresh water? Copepods, meron silang mga calcified coverings. Katong mga soft-bodied animals naman, mucus lang gihapon ang defense nila. And sa mga sa mga freshwater, the same thing, malaki ang tulong sa mga unicellular organisms, yung vacuole, contractile vacuole. Sa paramecium and amoeba, since na-expose man sila sa mga freshwater environments, ang contractile vacuole ang naga-expel og excess water. Mo na dili sila basta-basta mubuto. Unless kung mo silang submerge sa asido, mamatay yun na sila. Mamatay po na sila pag sobra po ka salty. So, kay murag dili sila suited in that kind of environment. Hindi mag-work. So, this is a paramecium. One of the known protozoans. And sa loob nito, you have the contractile vacuole. I think ang gigamit na microscope nito is SEM or katong another type, pero makita gihapon live man kasi when we use SEM dapat patay ang organism. So mo ni ang highly detailed structure ng vacuoles sa paramecium. So, water balance of cell within walls. So, the cell wall helps maintain water balance. So, it makes their shape turgid or it prevents them from bursting. So, in plant cell, hindi pumutok ang mga cells when exposed to hypotonic solution. So, asa to? Muna ang advantage sa mga plant cell. And there are also some protozoans na may cell wall. Hindi cell wall, lahi ang composition siyang cell wall. Naiuban like katong mga rotifera and foraminifera. Ang foraminifera likely related sa mga amoeba, pero kaning foraminifera naa sila'y calcaneous covering. Ang mga rotifera naman is murag naa sila'y silica, and even katong mga dinoflagellates naa sila'y mga silica that covers their others mo na mag-shrink at the same time dili pud sila mag-burst. So intact ang ilang shape. Facilitated diffusion. So ganina, another type osmosis is another type of passive transport, and second type of passive transport is facilitated diffusion. This is needed by the help of certain proteins. So, muna isa sa mga functions sa proteins, aside from being a channel, pwede din silang agent. It facilitates the molecules to enter the cell. So, one good example of channel proteins, aside sa mga example ng facilitated diffusion, channel proteins pala. Channel proteins like aquaproteins and ion channels. So, facilitated diffusion, ito pa yung mga integral proteins. Ito yung tunnel, passageway ng mga proteins. So, we have here aquaporins that facilitates diffusion of water, and ion channels. Iba pala yung protein na magtulong sa isang molecule papasok doon sa cell. Iba yun. Okay, this is an example. Mga sodium-potassium pump. Ganito din. So, this is the carrier protein within the cell. And another carrier proteins. Carrier proteins is also facilitated pala. Carrier proteins undergo subtle change in the shape of translocate solute binding site across the membrane. So, pero kailangan when a carrier protein, may requirement yan siya. Dapat fitted sa shape and its affinity. Hindi din basta-basta ang molecule na pwede makapasok sa or sa carrier proteins. Mo po na selective. Asa na itong video? Aking arms.
In the process known as facilitated diffusion, a special carrier protein with a central channel acts as a selective corridor which helps molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid. Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process by changing shape and moving the molecule down its concentration gradient through the membrane into the cell where it is released. Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules down their concentration gradient, and this movement is carried out without any input of energy. However, in facilitated diffusion, the movement of molecules will only take place if it is facilitated or helped by a special protein carrier in the membrane. Facilitated diffusion can occur in either direction, depending on the concentration gradient. If there is a higher concentration of the particular molecule inside the cell, the same carrier protein would then transport the molecules out of the cell.
So, that is facilitated diffusion. See, how about yung active transport? So, opposite lang siya sa passive because when we say active, the energy. So, mag-expel, magamit na ng energy. So, ito yung some some of the active transport. So not all molecules also can enjoy the active transport. Like the passive transport, may certain molecules din ang makapasok. Like the passive, active transport moves substances against their concentration gradient, but it requires energy. So kung walang ATP, hindi siya mag-work. So, muna ay primary requirement ng active transport. So, ang protein na pang-active transport, hindi siya gagana kung walang energy ang nakatapat sa kanya. When once the ATP attaches to it, then makaano siya ng molecules. Makapasok at makapalabas. So, one example, sodium-potassium pump.
Recall that there are two types of cellular transport, passive and active. Passive requires no added energy, but active does require added energy. Active move of low concentration to an area of high concentration or up the concentration gradient. This is the opposite of passive transport, and because it's more difficult, it needs the energy molecule called ATP, which stands for triphosphate, because there's an adenosine molecule. There are three types of active transport: membrane pumps, endocytosis, and exocytosis. Let's look at membrane pumps first. Membrane pumps are just carrier proteins that move substances from areas of low concentration to areas of high concentration. Let's look at the sodium-potassium pump to see how it works. Outside the cell, there's a high concentration of sodium ion, or Na+. High concentration of potassium ion, or K+. The cell has ion channels that will concentration. So the sodium-potassium pump more sodium out and more potassium in all the time. Let's see how this works. First, 3 sodium ions from the cytosol bind to the protein, and a phosphate group from ATP breaks off in the carrier protein. This provides energy for the shape of the protein to change, and the sodium ions can be released into the outside of the cell. Now 2 potassium ions will enter the pump, which causes the phosphate to break off and return the pump to its original shape and release the potassium into the cytosol. Again, sodium will diffuse into the cell on its own, and potassium will diffuse out. The pump forces the sodium to be outside the cell and the potassium to be inside the cell. Another way that cells work is through vesicle movement, either endocytosis or exocytosis. Endocytosis brings items into the cell, and exocytosis pushes them out of the cell. Endocytosis is the process by which cells ingest external fluid, macromolecules, or other large particles, even other cells. We can be more specific about what materials undergo endocytosis. Phagocytosis is where large particles or whole cells are ingested, and pinocytosis is when solutes or cell drinking. Here you can see the way it works. There's a vesicle which can carry the substance to any part of the cell.